We have a podcast about energy.
The jewels that these national labs are.
In terms of science and scientific capabilities.
Big dreams can happen.
Keeping our nation safe.
Clean energy is the wave of the future.
It's America's economic engine.
It's science
for the people.
This is Direct Current.
Hey everyone, welcome to Direct Current. I'm Matt Dozier.
And I'm Court Creer.
We've been hard at work on new episodes of the podcast, and we've got a fun one for you today.
So, Matt, you went to a big conference in D.C. to talk to some scientists recently.
Yep, AAAS. That's the American Association for the Advancement of Science. And they have this huge annual meeting where scientists from all over the world get together to share their findings, discuss research techniques, talk about how to communicate science, that sort of thing.
Yeah, and there's tons of folks there from the Department of Energy and our 17 national labs.
Absolutely. And so I got a chance to sit down with a couple of them to talk about a subject that I find really fascinating: microbiomes.
Microbiomes. So like biomes, but super small? Kinda?
Kinda. There's this invisible world of microbes that live all around us, on our skin, in our guts, and even in the ground under our feet, and we're only just beginning to understand their function. The researchers I interviewed are especially interested in using genetic data to study the microbiome of soil. Turns out these tiny organisms can help us answer some really big questions.
Sounds awesome. After the break, we'll listen in on that full conversation recorded live on stage at AAAS 2019. Stick around.
Hello and welcome, everyone. This is Direct Current and Energy.gov podcast. My name is Matt Dozier. We're here at the AAAS annual meeting at the SciMic podcasting stage. I'm with the U.S. Department of Energy, and I'm joined here by two scientists from the Joint Genome Institute. We have Dr. Susanna Tringe and Dr. Jason McDermott from Pacific Northwest National Laboratory. Thank you both for joining me today.
Thanks for having us.
I would like to have you first just give us a little introduction, tell us about yourselves and your research. Susanna, I want to start with you.
Yeah, sure. I work at a place called the Joint Genome Institute, which is a user facility that does genomics work for the user community. And my own area of expertise is called microbial community genomics, using DNA and RNA sequencing to study whole communities of microbes. And I particularly focus on wetland soils and agricultural soils.
Yeah, and I'm from Pacific Northwest National Laboratory, which is in Richland, Washington. A lot of people confuse that with being in Seattle, but we're actually on the east side of the state. I'm a computational biologist. I'm a microbiologist by training, and I've kind of gotten back into microbiology through working on soil microbiome projects more recently.
Great. Well, appreciate you both joining me here today. We are here to talk about microbiomes. And I think just for basic background, could you help me understand what a microbiome is?
Yeah, sure. So microbes are found in all kinds of habitats, ranging from our own bodies, in our guts, and on our skin, to environments like soil or water. And usually there's a whole spectrum of organisms, not just one microbe, but a whole community that works together. And so the microbiome of soil or your gut is sort of a community that's thought of as a unit that works together.
There is a lot of talk these days, a lot of attention paid to the human microbiome, as you said, with gut microbes, that sort of thing. Now, your work specifically is more focused on soil, right?
Right.
So why study the microbiome of soil? Isn't dirt just dirt?
So there's actually a lot of important processes that take place in soil that relate to carbon and nutrient cycles and supporting plant growth or the animals that live there. So we're really interested from the energy perspective in how soil is involved in the production and uptake of greenhouse gases.
Right. Okay, so Jason, I want to get to your research in a bit, but first, like when it comes to the actual collecting samples and figuring out what is in the soil microbiome, how do you actually go and collect those samples, Susanna?
Yeah, well, in the wetland projects, we have a corer, so we can go out and take these cores of soil, usually just maybe a foot or two deep, and then we take that and usually mix it up and freeze it to take it back to the lab, where we can then extract DNA for sequencing or RNA. Another thing we do with the cores before we extract the nucleic acids is actually measure their ability to produce and uptake greenhouse gas. We have an instrument that we can actually put the core in and measure its production in real time so that we can really see that that piece of soil is carrying out this activity, and then do our molecular assays to see how that matches up.
So you're not just using basic like gardening tools, right, to collect these samples?
Yeah. You have specialized corers, and you use lab equipment to, when you want to keep it clean and package it and put it on dry ice and then get it back to your lab. You want
to keep the dirt clean?
As best you can, especially you don't want any of your human-associated microbes getting mixed in with your soil, and they're all over you, so you do want to prevent that.
Right, so it's actually pretty clean work when it comes to collecting and transporting the samples.
Yeah.
So tell us what you do with these samples once you get them back to the lab. So what is the goal here, and how do you do that work?
Yeah, so the soil samples, in order to extract DNA, there's a series of chemical steps that works amazingly well with a variety of samples. That the protocols have been worked out such that you can take it. You usually add some beads to break all the cells up, and you vortex it, which is kind of shake it really hard, and then put it through a set of purification steps that take all that soil and dirt away, and then take out all the proteins, and if you're looking for DNA, get rid of the RNA as well, and finally end up with pure DNA that you can use for sequencing.
Gotcha. So, and Jason, maybe you can jump in here too. So this data that you've collected, once you've sequenced all of the genes in this sample of whatever it is, whether it's soil or something else, what does that look like, to you in terms of the sheer, quantity of data?
It's a pretty overwhelming amount of data. If you think about, what you might open up in an Excel spreadsheet or something like that, you can think of that multiplied many, many times over. And it's something that you can't really deal with the kinds of tools the kinds of normal bioinformatic tools that exist. And so you have to use, in some cases, high performance computing. So that's like big compute clusters to work through some of the data, especially from samples like soil, which are very complex. You end up having just a lot of data there. I should also note on Susanna's, she said they take out the proteins and to discard them. At PNNL, that's one of the things that we focus on, is taking those proteins and some other kinds of small molecules and also characterizing those to kind of layer on top of the metagenome kind of information that's coming out.
And I think it's important to define that term, the metagenome. There's the genome. So this is, you're talking multiple genomes, right? I think people might be familiar with the sort of personal at-home genome tests. So is this anything like that?
Yeah. I mean, the basic technologies for sequencing are similar regardless of the application, and that although many of the technological advancements have been focused on producing cheaper human genomes, the whole rest of the field of genomics has benefited from those changes in technology. In terms of the metagenome, that is sort of a coined term using that prefix meta in the same way you might talk about a meta-analysis being a combined analysis of many data sets. The metagenome is the combined genome of many different organisms present in a sample.
Right. So once you have all that data, you have the, massive, super spreadsheet of all the fragments of genetic data. How do you go about making sense of all of that information? Whoever wants to take that, I'm sure there's a lot.
Yeah.
I mean, it's a really difficult problem, and one of the problems is that if you look at samples like human samples or something, the genes of humans are fairly well characterized, and we pretty much know what the functions of most of them are. With a metagenome from a microbiome, we have lots and lots of protein coding sequences. So these are the bits of genetic code that actually do code for things that do work. And we really don't have a good handle. I mean, some of the estimates are as high as 80%, 70 or 80% of the pieces. We just don't know what they do. And a lot of them are not similar to anything else that we've seen. And so it's a difficult process. So the first pass is probably just to, match things up with things that have already been seen. So you can kind of go through and say, oh, this is definitely a certain kind of bacteria that we've seen before. You know something about what the bacteria does. And then you kind of go down the line of like, okay, here's a bunch of stuff that we know what the function is, but we can't really attribute it to any particular organism. I mean, it comes from a particular bacteria, but we don't know where that comes from. And then there's this whole bunch of dark matter, which is relatively unknown. Like, we can look at it with the current tools, and we just don't know what it is.
And you're using dark matter kind of as a euphemism there, right?
It is definitely a euphemism, and so what I was using it as is basically just the unknown genetic potential of microbiomes,
right,
in that case.
So, Susanna, specifically in soil, we were talking earlier that basically we don't fully understand what soil's made up of. Is that generally correct?
Yeah, and a lot of the organisms there are organisms that we are not able to cultivate in the lab, and the way we've learned that they are there is through these molecular techniques. We can see the same sequences showing up in our DNA sequencing, or at least the same families, and we can look at particular genes that help us place it on a sort of phylogenetic context, but ultimately they might not be at all closely related to the things that we've grown and sequenced and know what they do. Right. Yeah.
So in say like a typical sample of soil, of all that information that you collect and get, I mean, how much of that can we actually interpret at this point?
Something like less than half, that even if you can, depending on what you consider interpret, you'll take your data, you'll clean it up, you will try to assemble it into genomes if you can, which for a single genome would always be the objective, to get a good quality, assembled genome, so not just each individual read, but how they all go together, and then annotate genes, these protein coding sequences on it. And we can, for the metagenomes, it's not that hard to say this is probably a protein, because it could code for an amino acid sequence. Often you can say it's a protein, and it's similar to something else that's been sequenced some time, but the fraction that you can actually say it's similar to a protein that someone has tested in the lab and carries out this function is, I think, maybe 40% or something of the proteins that you see there.
Right, and you said that a lot of these microbes in the soil, we can't cultivate them in the lab, right? So we then can't really study them thoroughly. Is that basically right?
Yeah, exactly. What you'd love to do is test your hypotheses. Say, I think this organism can eat this particular carbon substrate, and what you'd like to do is grow it in the lab and feed it that carbon substrate and see that it takes it up and does something with it. But in actual practice, since you can't even get it by itself, you don't have any way of testing that kind of hypothesis in a simple, straightforward way.
Right. Okay. So, with all of these unknowns, with all of this data, so, what are some of the challenges and some of the ways that you try and tackle, sorting through, comparing all this data and figuring out what it means?
Sure. Well, one of the most useful things that comes in is really having a great reference database, because like we said, the first thing you do is compare it to everything else you have and try to say, are there other things that are close enough that I can kind of jump that gap and say, I think that it carries out this function. And so having as many genomes of just isolated microorganisms as possible really helps us interpret it. But also being able to compare the whole spectrum of genes that you have present in your sample with what people have seen in other samples that are hotter or colder or wetter or drier can really help you sort out, there's these thousands and thousands of genes, which ones might be the important ones in your particular system. Right. So the comparison is really the most powerful tool we have for these data that we have a hard time interpreting.
Right. And Jason, that's something you're involved in, right? With the, comparative analysis across, these databases and that sort of thing. Is that right?
Yeah, that's true. I mean, we're working on ways to extend some of the existing methods for looking at similarities. And so there are very good ways of identifying similar sequences. So this is the gene sequences or the protein sequences to tell you what this unknown protein is similar to. But like I said, there's still a lot of territory, and there are a number of cases where we know that the proteins have the same function, but we know that their sequences are very different. And so how do you kind of try to take those existing methods and extend them so we can get, more insight into what's actually going on?
Right. With so many unknowns, does it ever feel, frustrating or overwhelming to just look at all this data and say, what does it all mean?
I think it's really exciting, actually. I mean, yes, there's an element of frustration in that you'd like to push further, and sometimes you have things that you're like, oh, I really want to know, more about this system. But it's really interesting to think about the potential of this data and the things that we don't know. We're working on at PNNL on a project where we're trying to characterize some of the viral sequences that are in this. And so viruses actually infect bacteria, and there's a whole class of viruses that infect bacteria, and we can identify those from those metagenomes from the microbiomes, but we really have very little idea of what's going on with those, why those viruses are there, and what they're doing in terms of the dynamics of the microbial communities.
So that's even beyond the microbiome. That's the virome, right?
The virome, yeah.
So you touched on things we can do with this and exciting opportunities. So Susanna, tell me a little bit about kind of what you're trying to learn from this research. What are some of the big things that you're able to study and kind of shine more light on?
Yeah, there's a lot of different things we're trying to learn, but one of the main motivations behind my wetlands work is really not just better understanding the carbon cycle, but identifying better ways that we can help shift the greenhouse gas balance in the atmosphere. And so wetlands in particular are known to be very effective carbon sinks in that they have vegetation that takes up a lot of carbon dioxide and converts it into plant matter, but they can also be greenhouse gas sources. They produce methane at times or just CO2 from breaking down the plant matter, and that's a totally microbial process. And so there's potential for it to be a carbon source or sink. And when that is the case is not that well understood. There's sort of general trends, but any given wetland, it's hard to know, where it's going to fall on this spectrum. And there's a lot of wetland restoration going on now, usually aimed at improving habitat or water quality or flood control, not usually towards carbon sequestration. But I think all of those wetland restoration projects have the potential to be carbon sinks, but only if we really know how we can make them carbon sinks and not carbon sources. And so we're trying to look at the microbes and say, this sample produced a lot of methane, this one's not producing much at all. What are the biogeochemical and genetic factors that we can correlate with that pathway? And then can we step back to what was the environment that made that conducive to that particular balance.
Right, and so for your wetlands work, I think some folks might be, familiar with the sort of general area if they know the Bay Area at all. So tell us a little bit about kind of where you've been doing that research.
Yeah, and so we're located in the Bay Area, so it was a perfect place to study wetlands in this Bay Delta region. And our initial studies were up in the Sacramento-San Joaquin Delta, where there used to be a whole range of wetlands, but almost all of it has been converted to other uses, mostly farmland, because they have very rich organic soils that make great farming land. But unfortunately, that wasn't a very sustainable process because that organic soil tends to break down once you've drained it. It is metabolized into carbon dioxide. It gets compacted by the farming equipment. So if you go out to these islands, it's very striking that they're very far below the surrounding water and prone to flooding, because if the levees breach, they become flooded. So the wetlands we started studying there were partly to try to build back that subsided land. And then we've also been, in terms of studying this whole question of carbon cycling, we've been sampling wetlands throughout the Sacramento Delta and the San Francisco Bay region, and most recently the salt ponds in the San Francisco Bay, many of which are slated for restoration. And so we gave us an ideal opportunity to compare unrestored and restored
And I think I've seen those from the airplane.
Yeah, they're very striking. They're very bright red because they have these halophilic microorganisms that produce red pigments.
So kind of moving on from that, what is for each of you the most exciting thing about your research that you're working on right now?
I think for me, it's really the potential to have an impact on our understanding of the environment and our stewardship of the environment, being able to improve the way we manage our ecosystems, but also just this incredible power for discovery that you sequence almost anything out in the environment, and there's still a whole bunch of it that's not understood, and that can be frustrating. But you also think, imagine if we really did know what all of this was, that there are probably some amazing discoveries just lying in wait if you can figure out how to interpret the data.
Right. Yeah, and that's really exciting for me as well, as I expressed. I think one of the key parts of that we're really interested in, and I know JGI is interested in as well, is kind of the open science idea, which is trying to make as much of this data available and usable by the scientific community, because we have specific questions that we're asking about it. But if we can put it out there and have other researchers come and ask different questions, they're going to get, really interesting discoveries that we never even thought of. And so, part of that is moving that data forward and putting it out so that we can, enhance the biological discovery. I'm really excited about how we take different kinds of data. So the metagenome data, but also, as I mentioned, we have protein measurements that are kind of similar, and small molecule measurements, and how we knit those back together into a picture of an entire microbial community. And you can kind of think of it like this is a very interdisciplinary kind of approach, like your modern medicine, where you go in and you see a doctor, but you're not just seeing that general practitioner. You're seeing an X-ray technician, you're seeing a nurse phlebotomist, you're seeing, a bunch of different people with a bunch of different expertise. And how do you take each of those pieces that you get from that and knit it back into, a diagnosis of your problem? So that might be, how do you keep carbon in a form that's not in the atmosphere, or things like how do we enhance agricultural production, which is something that we're interested in from our soil work.
Sure, biofuel crops.
Biofuel,
yeah. You mentioned briefly the importance of partnerships. Obviously there's the interdisciplinary work, but also the open science, bringing other researchers, other institutions in to kind of help tackle this enormous challenge of interpreting all this data. Tell me a little bit about that and the way that sort of the DOE and the national labs have been working to facilitate this work across disparate sites and institutions.
Yeah, well, like you said, some of the data can be overwhelming, and that means when you're doing your analysis, you have to really go after a question, but then someone else may have a completely different question that will be answered by your data that they will approach in a totally different way. And some of the incidental discoveries have been really amazing. Like these CRISPR pathways were first discovered in microbial genomes, and that was using data from sequenced genomes, and no one had to generate new data in order to discover that pathway. And so we've tried to always make our genomic data public, and I think now with all of these other types of data, similarly, you might integrate them in different ways. You have these metaproteomics or metatranscriptomics and metagenomics, and having all of those available in one place where you can pull them in and come up with creative strategies for analyzing them is really powerful. So there's an initiative by the DOE to develop environments where you can do better analyses, as well as databases that store all of these omics data and allow you to retrieve them and compare them in the ways that you want to.
Yeah, and this takes a lot of coordination across sites with different expertise. We're certainly partnering on our soil project with JGI to look at the virome, as I mentioned, but in some other cases as well, just utilizing the data and the resources that they offer.
Yeah, and it's also worth noting that both EMSL and JGI are user facilities, and so we're not just doing the sequencing or characterizing or looking at the samples that we find interesting, but we're putting out calls to the user community to come use our capabilities. So we have, the JGI has a program called the Community Science Program that's just open to anyone doing research in energy and the environment in the DOE mission spaces, and they can come to us and work with us to use our capabilities, and the same with EMSL.
EMSL is the Environmental Molecular Sciences Laboratory, and it's co-located at Pacific Northwest National Lab, so it's very similar to JGI.
And I'm sure there are folks here at AAAS this weekend that have been involved with both of your institutions as well.
Definitely. Yeah. And all of those data from those projects are made available to the scientific research public in this spirit of open science, so that those discoveries can really be leveraged upon each other.
And I think it's really important to note that those kinds of efforts have been driving forward the field. We're closely associated with EMSL, but we're not in EMSL. But because we're using those resources, all the data that we're making has to be publicly available, has to be open, has to be reusable, and so that's been a really great thing to be working with.
Absolutely. Well, thank you to you both, Susanna Tringe, Jason McDermott. Really appreciate you joining me today.
All right. Thank you.
Thanks very much.
Thank you to AAAS for having us here on the SciMic podcasting stage. This has been Direct Current, an Energy.gov podcast. I've been your host, Matt Dozier. You can find us at Energy.gov/podcasts or wherever you get your podcasts. And thank you all for listening. Thanks again to AAAS for having us. Thank you to the folks at the Joint Genome Institute, Pacific Northwest National Laboratory, and the Department of Energy's Office of Science for helping arrange the interview. To Kirsten Hofmackel at PNNL, who was supposed to join us but had her flight from Washington canceled at the last minute. And of course to Susanna Tringe and Jason McDermott.
As always, if you've got a question or want to leave us some feedback, email us at directcurrent@hq.doe.gov or tweet at Energy. And if you're enjoying the show, share it with a friend and leave us a review on iTunes. We read them and we listen. Direct Current is produced by Matt Dozier, Paul Lester, and me, Court Creer. I also create original artwork for every episode. You can find that on our website.
Additional support from Anne-Marie Horowitz, Ernie Ambrose, Gigi Frias, and Atik Waraish. We're a production of the U.S. Department of Energy and published from our nation's capital in Washington, D.C.
Thanks for listening.